is going to be talking about power and sample size. Dr. Martin Lee is professor of biostatistics and CTSI service statistician with experience in pharmacological statistics and clinical trials. He's on the faculty of Charles University and teaches statistics at Charles University and collaborates with investigators, especially in the areas of epidemiology and diabetes. In addition, Dr. Lee collaborates with the VA Greater Los Angeles healthcare system in several roles. He is a CTSI investigator. He's involved in teaching courses through to CTSI. The courses are accessible online. In addition, he works with BSD, CDM faculty to conduct interinstitutional biostatistics grand rounds and provides mentoring and service on Ph.D. committees. We welcome Dr. Lee. He's gave some excellent lectures at the first institute, so we're really going to treat to hear about power and sample size.
Okay, well, thanks a lot. Thanks a lot, Dr. Friedman. I appreciate that. Good morning, everyone, and I will try to make this as succinct and as interesting as possible. What we're going to talk about today, actually, the title of this hour is design, essentially design of studies. So we're going to actually have two separate presentations. One is about sample size and what goes into that. And then because this group is interested in community-based studies, where I'm also going to tack on a brief second presentation that's going to go into the particular types of designs that come about when we're trying to do things in the community as opposed to just within, let's say, a given institution and so on. Now, I'm sure everybody, the first question I always get as a statistician working on any project, and by the way, I've been doing this, I know you can't tell from my appearance, but I've been doing this for 42 years. And as I always like to say, yes, I was 5 years old when I designed my first study. So pretty amazing, I suppose. But the first thing that I always get when people ask me to participate is, how large a sample size do I need? And that sounds like a simple question for most people, and it is, and it's an obvious and obviously a critical question, but it goes a lot deeper than just sort of providing a number, because there are a lot of aspects that go into that. So what we're going to do today is talk about the parameters that go into these calculations briefly, and I mean very briefly. I'll show you some of the formula, although to be frank with you, the formula we can, sometimes that makes people just sort of their eyes glaze over anyway, and we can probably just quickly go through those slides, because frankly, there are in fact software that do all these nice calculations for us, as opposed to the old days when we had to do these all with a little desktop calculator. And I guess I am dating myself at this point. So what are the things you need to know in terms of the issues that go into the study that will ultimately dictate what type of how you'll do the calculations of sample size? And there are a number of things that you may not have thought about. A lot of studies we do involve randomization into two groups, and that for most people is the most important thing that we consider. But obviously there are going to be studies which we don't or can't randomize, and that's going to change things. Whether we use matching, now to make sure everybody understands that particular point, do we in fact try to pair people up, whether it's in order to make the two groups comparable, or it's a much more convenient thing to do, like for example in a crossover study where every patient gets, or every participant gets both or multiple study groups. That's those kinds of designs dictate different types of calculations than if we just simply take a group of people, randomize them in a very simplistic way, or just simply assign people to groups in whatever way we decide to do that, and not try to take into account any kind of matching algorithm. Clearly, the number of groups you have, most studies, of course, have two groups. Typically, it's a experimental intervention versus a control, but a lot of studies, have multiple groups, particularly if we're doing drug studies where we have multiple dose groups. But then often we try to look at different types of demographic groups in studies, particularly community-based studies. So that's going to matter. What you're measuring, all right? Now, we usually think of measurements in terms of two broad categories, whether it's numeric, which fancy name for that is quantitative, or it's categorical, yes, no type variables, or simply, you know, as they say, categorical variables. And that's a different type of analysis, and then therefore a different type of sample size. And then I'm not sure this group is going to do much in this particular type of data, but time to event. In other words, how long does it take for something to happen? And oftentimes that type of data is censored, meaning we don't get to know exactly what that time is. For example, when we look at how long somebody with a particular disease and gets hospitalized stays in the hospital, you'd say, okay, well, what's hard about that? Well, what if they die? Then we have no idea, because obviously they weren't unfortunately discharged alive. Do we take repeated measures? In other words, do we measure the same person multiple times? That creates more complex data, and of course we have to account for that. Do we include what we call multivariate adjustment, characteristics of the subjects? Do we incorporate that into the way we're going to look at the data? And then, of course, the age-old question of whether our goal is to show whether the intervention is strictly better or in either a positive or negative way, or are we just simply looking for a change in the effect of the intervention, that is both positive and negative. Now, we also need to know what our goal is. Now, most of the studies I'm sure you're all interested in are what we call hypothesis testing. You say, okay, this particular intervention or this change in the way you're going to deal with the individuals who are part of your population, we think it's going to work. And that's a hypothesis, of course. But a lot of times, or on many occasions, we're interested in just estimating an effect. For instance, given the world as we know it right now, we might be interested, for example, right now in estimating how many people are going to take the COVID-19 vaccine when it's available. That's, of course, a different type of problem. We want to know a percentage, and I'm sure you've heard many of the numbers bandied about by people who have done those kinds of survey sampling. And of course, that's a different type of statistical analysis and requires a different type of sample size calculation. And then we also have studies. Most of our studies, of course, as we've been indicating, are to show something is better or improved or decreases, for example, improves survival or improves participation in getting treatment or things like that, or reduces things. For example, reduces in diabetics, you reduce A1C or you have better glucose control. But there are some studies in which we're trying to show equivalence or just that it's no worse than what's being done. Why would we do that? That's simply because some therapeutics are intended, or some interventions are intended just to, for example, lower cost, make it easier, a different method of administration in terms of a drug. So we do studies like that too. So it isn't always showing better or improvement. It's sometimes just showing no worse than, in order to be able to introduce other benefits like cost, for instance. And then very, very important, this last bullet point on this slide, which is, what are you really trying to show? What we call the minimum clinically important difference, which is the term we use in statistics, or the symbol we use in statistics, delta. Okay, how good do you think this really is? Now, nobody ever knows, of course. We like to think we've invented something that will work, but we don't know for sure. But in designing a study, we need to say, how good do you think this is going to work? Because if it works at least that well, I think that will be useful in practice. And if it's a drug, it means I might change my medical practice and use that drug. If it's a community-based intervention, we might now change the way we do things as far as whatever that intervention is concerned in the community. But we have to have an idea, and we call call this again, a minimum important difference, because you have to have a certain benchmark, a certain minimum you would be impressed enough by that, if that were true, to change your practice. Now, with equivalence, it's a different story. We need to know, what do we mean by no, not different from one another, or no worse than, and that's a similar kind of concept, but in this case, it's the maximum difference that would make us clinically indifferent to either intervention. And so we have to figure that out. And that's usually done by sort of consensus by the people who are in the study. Now, the other aspect, so once we've got a delta figured out, let's say we're talking about a superiority trial, then the next thing, then many of you, I'm sure many if not most of you, have had at least some exposure to statistical methods. You know that in any kind of experiment, you're going to have the possibility of making one of two types of errors. We call these statistical errors. And errors is a bad term, but nonetheless, because it sounds like, well, we're screwing up, but it's not. It's just because in statistics, as I'm sure you're all aware, we can't make statements about the experimental results with absolute certainty, for the simple reason is we're looking at a sample, not the entire population who might be getting this, whatever this intervention is. So as you can see from this table, we make a statistical decision, as you can see on the left-hand side. We either decide to reject or not reject, or some people call it accept the null hypothesis, which is the statement that the intervention we're looking at, whatever it is, doesn't have any effect. So either we say, yes, it does have an effect, which means reject, or we say we don't have any evidence or insufficient evidence to say that it doesn't do that. So we would accept or not reject the null hypothesis. Now, the truth is that there's always a truth. We just never know what it is, but there's a truth in the real world. And that is either the intervention really does have the purported effect or it doesn't. So you can see if where the errors can occur. So if you have an intervention that really doesn't do what you hope it does, but you still conclude that it does have an effect, that's called a type 1 or alpha error. And conversely, if the intervention that you've come up with really does work, but you fail to recognize that, that's called a type 2 or beta error. What we're trying to do in designing experiments is control those two types of errors. Now, of the two, we consider alpha more important than beta, or type 1 more important than type 2, because a type 1 error is what we call an error of commission. We're saying something works, but it really doesn't in the long run. And so we really don't want to foist on the community something that really won't help the community, and you go to all that trouble, and maybe there are issues in changing practice, and maybe some of those issues can be deleterious, but you think that the risk to benefit ratio is positive. So a type 1 error is more important. And as I think you all are well aware, most people set the type 1 error, the level that you're willing to accept in any experiment, at about 5%. That's called the significance level, and so on and so forth. The type 2 error is less important because it's an error of omission. You're saying something doesn't work when it really does. And so people are willing to accept a little bit higher level of that type of error in designing experiment. So we usually set type 2 error as 10 or 20%. Now, a lot of people don't even talk about type 2 error. They talk about the opposite of it, a 1 minus beta, which is concluding something works when it really does. And that, as you probably know, is called power. So instead of talking about 10 or 20% type 2 error, we talk about an 80 or 90% power. So we need to choose those. And so we've just mentioned those. And so this slide just basically reiterates what I just said. So what's the effect? How does all of these different things interact with one another? Well, one thing that these two pictures show you is that the larger the difference you're looking for, delta, in other words, which is again how well you think this intervention is going to work, the larger the difference, the smaller the likelihood that you'll fail to see it. In other words, if you have an intervention that really, really works, the chances are you're going to be able to see it. But if you have an intervention that has a very minimal effect, It's probably a lot, it is a lot less likely that you would see it. So the top is a small delta and the bottom is a large delta, but we can mitigate that by sample size. So regardless of the delta, and here you're seeing essentially that top picture again, but you can see from the two pictures on the left is one sample size of, in this case it's a sample size of 25 in this particular experiment. On the right side it's a sample size of 100. And what happens is that the larger the sample size, even though the delta is still the same, you have a much higher probability of making the correct decision of rejecting the null hypothesis. So what we ultimately do is, I think, you know, what we're saying here is you pick an alpha, you pick a beta or power, you pick a delta, and then the sample size then results so that out of that calculation, so that you have a risk of missing the effect of 5%, a risk of or the probability of seeing the effect of not 80 or 90%. And that's what the sample size calculation gives you. And so that all goes into these calculations. So again, these are the different things we need. Now, with quantitative data, you also need to know what the variability is. Going back to that last slide, the variability of your data, which is represented by the, you know, by the spread of the distribution of results under either of the two hypotheses. You need to have some idea of that. And so that's also a challenge for us when we do these calculations. When I say the variability, I mean whatever you're measuring. Let's suppose it's reduction in a diabetic of their hemoglobin A1C. We need to know what the variation is from subject to subject, what you expect that variation to be, the standard deviation, in other words. And there's various ways to get at that, using the literature to see what other studies have seen, or sometimes using the rule of 4, which maybe many of you haven't heard of. The rule of 4 is a really simple and kind of nifty little rule that we often use when we don't have any literature and don't know what the variability is. We basically ask, well, for this particular outcome, what do I think the minimum and maximum values could possibly be for the outcome? In other words, what's the smallest difference, the smallest result I could find and the largest result I could find for, let's say, whatever that measurement is. And then you take the difference of those two and divide by 4. And surprisingly, that often gives you a pretty decent approximation of the standard deviation. So it allows us to get at this number without really having a whole lot of information. Now, I'm just going to roll through these because, like I said, I don't want to overwhelm you, because for a lot of people, this is like, okay, this is where I kind of close my eyes or sort of look the other way. It's like, you know, it's like seeing a bad accident. You know, you want to look, but you really don't want to look. These are the formula, for instance, for looking at the difference between two means. And all I can just point out is all the different things we just talked about are in the formula, and I'll leave it at that. Now, one thing, this is an illustration by just simply looking at some calculations, that you can see that the sample size dramatically increases. You can see it's an exponential, depending on how big of a difference you're looking at. And what we're defining here on the horizontal axis for this graph, is what we call the effect size. What that means is the difference between the means of your two groups, whatever it is you're measuring. So let's say we're looking at a study of reducing blood pressure. So there's the blood pressure mean for one group, the blood pressure mean for the other group, and how different we expect them to be. What we're looking at here is that difference divided by the standard deviation, and this is known as an effect size, sometimes known as Cohen's d, if you want to get really technical there. And what you're seeing here as the difference as a function of standard deviation. So what these numbers are on the horizontal axis are the number of standard deviation difference between the two groups. And as you can see, for you know, differences of as much as a standard deviation roughly or more, the sample size requirements are very, very minimal. And you'd say, oh, okay, that's great. Most of our studies are going to be small, but I'm here to tell you that most treatments, most interventions never show that kind of an effect. A standard deviation difference between two means is an amazingly large effect. What we usually end up seeing, average differences we see in a lot of studies, and there's been research surveys of this sort of thing, is about a half a standard deviation. And then as you can see, you can see that beyond that, when you get down to very small differences, the sample size requirements are huge. The good news is you really never want to do studies for that kind of an effect, that small of an effect, because remember what we said earlier, the effect that we're trying to demonstrate in a study represents the minimum clinically important or meaningful effect. So you don't want to do really large studies to show really tiny benefit, because nobody is really in a practical sense going to care. So the important message for you is design studies that are both going to provide the statistical significance that everybody, of course, aspires to, but also make sure that the study is showing a clinically significant effect. I talk about these two types of significance, clinical significance, statistical significance, and we try to design studies that meld the two. That's the key. So here's an example of exactly what we were just talking about: blood pressure. And in this particular study, it was looking at blood pressure, the effect of removing considering drug cessation in someone who's let's say addicted to cocaine, for instance, and we're trying to see in one group, in the two groups, those that get the intervention versus those that get some control, whatever these happen to be, what effect it has on the individual's blood pressure. And so you see here that there is a difference between the two groups in this study of about 5.4 in terms of the mean, 5.4 units in terms of blood pressure. And the p-value is quite large, it's 0.46. Now, if you thought that 5.4 was a big difference in blood pressure and systolic blood pressure, what we are seeing here is that this study was not powered. We didn't have a large enough sample size to detect this kind of a difference. The power going backwards to calculate it is about 13%. And you can see that the study size was 8 in one group and 21 in another. This is an actual study, by the way. So they weigh, what we say, the terminology we use is that they were way underpowered for this study. The effect that they saw, which they believed was important, was obviously not demonstrated to be statistically significant. Now, if we wanted to show that same delta and we wanted to redesign the study using the value of 5, roughly, is the difference between the means, and an average standard deviation between the two groups of about 17. And we wanted 80% probability of getting statistical significance if this delta really is the truth. We'd need 154 subjects. And so the original study was 29, so that means about 5 times as many people as the study set out to collect. So this is illustrating in a very, extreme way how important doing these calculations are in order to avoid doing a study, seeing the difference that you would hope to see, and yet getting virtually, clearly no statistical significance. And then, of course, the study gets ignored. And people then use the expression, well, your study was underpowered. We hate hearing that. We never want to hear that from anyone. So that's why these calculations are so important. Now, just to illustrate, this is an old version of a program that I really recommend, and I don't have any stock in this company. It's a fairly inexpensive program, and there are others, and I'll mention a couple others in a few minutes. But this is a program called PASS, or standing for power and sample size. And you can see how easy it is to use this program. You simply you put in the expected means that you would see in the two groups, and you're allowed to put multiple values. So you can see that the mean of group 1, which is our intervention in this particular example, we put in a value of 10. The intervention group, we might see a difference of 1, 2, or 3. That's why we put in means of 11, 12, or 13. We put in our standard deviations. which again we come up with, and then just push a button essentially, and then you get this kind of an output. And so you can see for different values of delta, and you can see on where the columns that are labeled mean 1 and mean 2, you can see those different possibilities. So you can do what we're doing here is what we call a sensitivity analysis. Let's play with this. Let's see what we can detect for different sample sizes. And you can see the sample sizes range all the way from 12, which is the biggest difference with only 80% power, all the way up to 133 per group with 90% power and a small delta of 1. So these kinds of programs allow you to play these kind of what if kinds of evaluations, and that also allows you to, you know, do the kinds of calculations we often have to do, which is, well, yeah, I'd love to do the actual calculation, but I only have money to do 100 people in my study. Well, this is the way you find out what you get for 100 people, if that's all your funding allows for. Now, if we're doing the same thing with equivalence, then the other thing that we change this around a little bit, it's still all the same parameters, except the delta now means something different. It's what we mean by equivalence. It means how different can the treatments be and still be considered the same if that's the type of study you want to do. And then, of course, you still determine an alpha, you still determine a beta, and of course, a standard deviation, and you just run through these calculations. When you have proportions, which is obviously what you're going to have when you're looking at something that is qualitative, in other words, categorical, then the idea of what we mean by delta or difference, and here we're calling it D, is just the difference between the proportions. So, you know, P1 might be your baseline proportion, P2 is what you think the you know, the effect, the improved proportion would be in the intervention group, and what we're looking at either is defining what P1 and P2 are, or defining P1, your baseline, and then defining what the difference from P1 might be by using the intervention. Let me give you an example. So here's an example of a smoking cessation study. You have two different drugs to improve smoking cessation. So you have drug A expected to have, you know, 70% of the people in the study may give up smoking after 1 year. Drug B, 60%. So what else do we need? We need the significance level. Here we're picking a significance level or an alpha 1%, for whatever reason, and 90% power. And then you run through the calculations and you can see that it's 675 people per group. And part of that is because, well, there's various reasons why the sample size is pretty large here. And that is, there's a fairly small difference, a 10% difference between the two, an absolute 10% difference. We're using a relatively low significance level and power of 90%. You vary these things, and you can see you can get this sample size down by picking different sample sizes. Here's one example, one illustration of how the sample size varies. It varies as a function on the left-hand side of your baseline. You can see if your baseline, that is your control group proportion, you know, you can see it varies from 10% to 80%. You can see what it does to your sample size. So where you're starting as a baseline matters, and then how different you're trying to show from baseline matters as well. And as you go across the columns, you can see the bigger the difference, the smaller the sample size. So that's not that surprising. We've said this before with means, and now it's also true with proportions. So, you know, the tendency is to say, well, I can do a study of, you know, 15 people per group, right? Like, for example, let's take a baseline of 10%, an improvement of 70%, so to 80%, in other words. Wow, that's great. I can do a really small study. But the thing to always remember when you're designing studies is it's very unlikely that any intervention, I mean, you can count on one hand the interventions that have had such a dramatic effect. Let's say this is you know, ability to get off, to stop smoking, for instance. Most interventions are just not going to have that amazingly dramatic effect. So we've got to be realistic in what we think the delta is. And then again, here's the screen from PASS. Very simple. Again, all you have to enter is things like the effect size, the alpha and the beta, and then you get an output like this. And again, illustrating how different values of your baseline, different values of the delta, and different values of power will change sample size. And then, of course, we have a formula for equivalence, which we'll just mention in passing here. And then sometimes we do studies with multiple cases per control, particularly, when you're doing a case control study, for instance, if you're doing an epidemiological study where you have, say, an outbreak of a disease or an outbreak of some unusual event in a neighborhood, and you want to determine what the cause might be, you're going to go out and look for, matched people that have similar characteristics to the individuals, let's say, living in that geographical area, and then to see whether this has anything to do with locale or in famous case like the one in Central California from 30, 40 years ago, where drinking the water caused a very unusual type of cancer to break out. We want to, consider how many controls, how many people in the control group do we need for each case. And so there are formula for that. And what we find is that increasing the number of controls does improve power, but then it sort of tapers off. And so what we usually tell people, you never really get much benefit in having a case control study with many more than 3 or 4 controls per case. It just doesn't help you that much, and therefore costs the time and effort to find those people. Now, a couple of other issues that we want to just mention here in passing about this in our discussion of this, and that is there are fundamentally there are 2 different types of analyses we do with experimental data. What we call intent to treat, which is everybody that gets in a randomized study, everybody that gets randomized, put into the study, versus those that really complete most, if not all, of the study, which is known as a protocol analysis. So we have, as you all, I'm sure, aware in your experiences, you have people who drop out or don't complete the protocol or don't take all the drug. They may stay in the study, but they fail to adhere to what you wanted them to do. So we need to adjust our sample size for what we would refer to as missing or incomplete observations. And there's a fairly simple formula for that. This is a fairly, I won't say it's conservative, but it's a standard formula that people use, which is to take P, which is your missing or incomplete rate. So let's suppose P was, say, 20%. And what you would then do is take that sample size that you've just nicely calculated and divide it by 1 minus 0.2 or divide it by 0.8, which means you have to increase your sample size by 25%. 1 over 0.8 is, of course, 1.25. So you got to inflate your sample size to allow for the missingness or incomplete rate, or the dropout rate, as we usually refer to it. How do you know what that's supposed to be? Well, that's a guessing game. You don't want to assume it's going to be really large, because if in fact it turns out to be that case, people are going to question the study or the intervention, because if that many people are dropping out, maybe this isn't going to work in real life. So I think the standard number that we often use in our designing our studies is something like 10, 15, 20%, something like that. And of course, then it's incumbent upon us as investigators to try to keep that at that type of rate. Now, I've mentioned the software. I was showing you screenshots from past 2008. I haven't updated the slides from this for a while. The version that we're on now is 2020, and it's an amazingly easy program to use. There's also another excellent program that was invented by some folks at UCLA, and it's called nQuery. I haven't used it, but I hear lots of good things about it. And there are also a lot of internet calculators. You can find websites where you just plug in the numbers and you get a calculation. The only difference between buying a program which comes with validation and it's probably got certification that could be used, let's say if it was an FDA submission. And internet calculators is, well, the latter you're really not as 100% sure of, so you need to consider that. Now, what about estimation? Well, we have different kinds of formulas for estimation. Basically, now all you're interested in is ultimately with a mean or a proportion. Let's say, using my example from before, the proportion of people that are going to, that say that they're going to get vaccinated with, one of these new vaccines. Now, what you're going to do is you're going to create what we call, as I'm sure you're all aware, a confidence interval. So in determining sample size here, you need to have essentially two things. Well, first, three actually. You need to know what confidence level you want. Most people pick 95%. That's what we've all pretty much been, learned and used, as a standard number. And then with proportions, you've got to kind of take a guess at what you think the number might be. You've got to be in a ballpark. Now, as you say, well, how can I know? I just don't know. So the rule of thumb is to use 0.5. That will give you the maximum sample size in that formula. So if you use p equals 0.5, that will be the maximum sample size you need, regardless of what the true proportion or the proportion turns out to be. And the other thing is you need to know what we call the accuracy of the estimate, which most people know as margin of error. Okay, so we need to pick a margin of error, and if you've ever read lately in newspapers leading up to the election, they always talked about margin of errors for these election polls of 3%, 4%, 5%. So you just simply pick a number. What that means, that's the plus or minus that you create when you get to the actual proportion from the sample. That's the plus or minus that gives you essentially the breadth of your confidence interval. So you can decide how precise you want to be in that regard. And so obviously, the larger, the better the precision, the more the sample size. Similarly, with means, it's a little different because now you're looking at a quantitative number here. You need to know something about the standard deviation. We've talked about how we get that, and again, a margin of error as well, and a confidence level. So here's a couple of examples involving two different studies. One on the right-hand side about recidivism after treatment for cocaine addiction. We're expecting it to be about 20%. And so we want to do a sample to find out what the true recidivism rate is in the population. And so if we want a, let's say, a margin of error of 5%, about the purported 20% that we expect, you can see you need 246 people. In this other example, we want to know in a VA population the number of drinks, alcoholic drinks consumed on the average, and we want to estimate that to within plus or minus 2 drinks of the true mean. And we guess at the standard deviation or try to figure out what it is from whatever source we have. And you can see this is a much smaller sample size because the margin of error here at plus or minus 2 is actually pretty large compared to the standard deviation. So you get an idea how these calculations work. Okay, so that's sample size. Now I want to take you to a second lecture or a second portion of this lecture, which is the issue of design of community-based interventions. And this will include a brief mention of the issue of sample size as well. And how these types of interventions in the community are a little different than just doing the kinds of studies we've been talking about so far, which are, I've been alluding a lot to drug studies or interventions on a per individual basis. Now, we all know that a lot of the interventions that we look at, and when we think about community-based or healthcare provision and changing the way, obviously at the individual level, for individuals to change, let's say, health practice or taking care of themselves. We're going to implement these at a level above the individual, the person, the individual in the population. We're going to implement these at a provider level, at the doctor level, for instance, at a clinic level, at a hospital level. I mentioned something called a VISN here because this part of the lecture was originally being given to the VA, and VAs are organized by what are called VISNs, and that's on the one hand. So we're implementing the intervention like, I'll give you a couple of examples in a minute, but one of the ones that I'm going to mention is about smoking cessation. The idea of improving smoking cessation, we want, of course, individuals, in this case, this was at the VA, so we want veterans to stop smoking, but at the same time, we're going to change the way that information is delivered at the provider and institutional level. So we're measuring the outcome at the patient level. Obviously, we want an individual to quit smoking, but we're implementing at a higher level. So this is often known as a hierarchical design, or we have what's called hierarchical data. And the randomization, if we're doing randomization in this type of study, takes place at levels other than at the patient. Now, there's a few reasons for that. One is, if you were, for example, providing an intervention, explaining in a new way to an individual why they should quit smoking, and these individuals are, going to the same physician, and this physician needs to, well, this person's, let's see, this person is in the intervention group, so I got to explain something different to them than I do to a control patient who's also seeing me. You can see how it's very easy to be there will be contamination. Something about the way the intervention is delivered can get into the interaction with a control patient. It's also difficult, you can see, obviously, in just the way it sounds so convoluted to do that, it's difficult to implement multiple interventions at either the provider level or even at the institutional level. And it also turns out that some institutions prefer a particular approach. So maybe we don't even randomize. Some people say, well, this is the way we're doing things, and that's the way we're going to do things at this place. And other institutions are more amenable to changing their approach. So you have, obviously an easier thing to do is just change the practice of the entire institution if they're willing. So the problem, though, is that because you're now using the institution, for argument's sake, as your intervention, the basis for your intervention, you have small numbers. Even though in that smoking cessation study I've been alluding to, there were 10,000 people in it, there were only 20 institutions, and it's that 20 that matters, not the 10,000. So here's some examples of trials that have used this kind of, methodology. Trial done in '95, rapid early action for coronary treatment, getting people to seek medical attention when they, feel or someone notices someone having an MI or a heart attack. The intervention included a mass media campaign, making people alert of symptoms or, things that are leading up to the potential for having a heart attack. Well, you can't have a mass media campaign for some people and not others in a particular geography. So they had to randomize the participants by city. A nutritional education goal. This is something that you're all familiar with. A very interesting study in which the goal was to figure out whether putting calories or in this case labeling whether certain foods were high salt or high fat on the menu. Okay, could you do a study like that? Well, you can't give menus out, randomize people, and then some people at the same table get the menu with the information on it and some don't. There's clearly going to be cross-contamination. So they randomized by restaurants. And then this particular study I've been talking about, which was known as the QUIT study. By the way, there's nothing like a cute acronym for a study. If you're going to do a big study, first thing you got to sit around, and I kid you not, is figure out a cute acronym for the study. And so this was the one that was done at the VA, and we randomized by medical center. Now, group randomization is otherwise, or similar to, or otherwise known as cluster or hierarchical random sample. Well, it's similar to that, or randomization, of course, is not sampling, but the idea is the same in terms of what we're trying to do. And so the data itself is not a simple random sample. That's very important. The individual, because we're measuring things at the individual. For example, in the smoking cessation study, we're measuring whether the individual quit smoking at 1 year. But that data is not a simple random set of data, because the sampling unit is not the same as the analytical unit. The unit of randomization is not the unit of analysis, and that has implications for sample sizes. We'll see in a minute. And sometimes we worry about, even if we don't design a study this way, even if you do randomize at the individual, you might worry about whether, in a multi-center study or a investigator study, you may worry about whether people who go to see a particular doctor or go to a particular clinic or a particular hospital, their data is more alike than data from other hospitals. And so you worry about this so-called clustering effect, even if the design didn't actually account for it. The other important issue is whether or not you take repeated measures. Do you just measure? Oftentimes in many studies, we just do a pre-post type of design where we measure before and measure afterwards, otherwise known as a panel design. But oftentimes, and very frequently, we're gonna look in on the patients and the subjects many times. This factors into the design, and we have to think about it. Now, let's talk about sample size, because this has been kind of the theme of our lecture here. One of the easiest ways to think about sample size is to think about this so-called inflation factor that is engendered by clustering. Okay, what I mean by that is people who are in a, let's say in a particular institution, or let's make it simple, that go to see a particular physician, their data tends to be more alike than people who are at other institutions or seeing other physicians, and this has to be accounted for. Now, there's a really great book. I don't know if it's still in print, but it's probably the definitive book in my estimation. It's written by two Canadian statisticians, Donner and Clark, and this explains a lot of this. So I recommend that as a reference if you're looking for something. Just Google Donner and Clark's book on cluster randomized designs. Now, what this inflation factor is, and I'll try to explain this real simply, is this is a factor that you have to basically take your sample size calculations that we talked about in the previous lecture and inflate them by this number. Now, what is this number? It's 1 plus, and now you have this little factor here. What M bar is the average cluster size. How many people are seeing on the average a given physician? And then you have another number, which is the Greek letter rho, which is known as the intra-cluster correlation coefficient, which means basically how correlated are the people within a given cluster. This is typically impossible for the average person to figure out. So we take guesses at it. Now, how do we take guesses? Well, there's a lot of literature now. When I first started doing this about 20 years ago, there was no literature. And we were looking at this and say, well, this is a correlation coefficient. Now, we know correlation coefficients range, most of us are, we're familiar with the typical correlation that goes from 0 to 1 or 0 to -1. These kinds of correlations don't meet that kind of range. Usually these correlation coefficients, these intracluster, are in the order of about 0.05, 0.1, 0.2 is really strong correlation as far as this is concerned. So all the advice I can give you if you're starting to work on something like this is to kind of look for other studies in your particular arena and see what has been reported. But most of the time we get away with numbers like about 0.1. That's relatively conservative. And so when you take those, you do this simple calculation, 1 plus you know, the cluster size -1 times the correlation coefficient. You simply inflate your sample size by that. Now notice two important things. If m equals 1, if your cluster size is 1, of course this inflation factor disappears. Well, what does that mean? It means you're not clustering. It means you're doing a traditional design. And if the correlation is 0, same thing. You're using a you have a situation where the clustering really doesn't matter. Sometimes what people do is they try to get some pre-data. They try to go out and get from whatever source they can, maybe medical records or whatever, and see if there's any kind of correlation, say Dr. Smith's patient population, and see if there's a correlation in whatever outcome they're interested, and say, oh, it doesn't look like there is, so I can ignore this. I don't think that's a really good idea. I think if you honestly believe that clinically, you should incorporate it into the calculation. Notice that what really matters here is not so much rho, because rho, as I said, is going to be small. It's your cluster size. It turns out that the bigger the cluster size, which means a lot more people, right? If you have, say, 10 institutions and 100 people per institution, that's 1,000 people, right? That's going to, you can see the larger the cluster size, the bigger the inflation. So what we try to do, if it's all possible, is to have more clusters and fewer people per cluster so that this inflation factor doesn't get out of hand. So we've talked about how we get these values. So you can see here's the formula you saw earlier where, you know, the business part about it is the sample size for comparing two means, and then you can see the inflation factor is given there. So what you calculate is your sample size, and since you know roughly, or you expect certain, say whatever m is, 10 patients per investigator, per institution, then you have your sample size, so then you can back calculate how many institutions you need or how many investigators, because K is the number of clusters here. And so you're using the formula with an assumed number of people per cluster to back calculate the number of clusters.
Martin,
yeah.
You wrap it up and maybe have time for 1 or 2 questions.
Yeah, I'm just about done. So these are just illustrating the point about clusters versus number per cluster, et cetera, et cetera. So only thing I wanted to just show is how does that work? How does that come into play when you do the actual analysis? You can see here's a t-test, and you can see where the inflation factor gets incorporated into the denominator of your t-test. And illustration here of a simple study, and it turned out that in the study, if you fail to account for clustering in this examination of income between homeowners and renters, you would have gotten a significant result, but a very significant result. But by incorporating the clustering, you don't see significance at all. So the moral of the story is, and this is my last slide, the moral of the story is, if you fail to incorporate clustering, you may see spurious significance. Very important point. And then there's various software, Stata, SAS, there's a specialized program called Sudan, and another one called HLM. They're pretty hard to use, but if you're really into this sort of thing, it's important. Okay, that's basically it.
So we have time for 1 or 2 more questions. 2 questions.
I've overwhelmed everybody.
Yeah.
With my magical
I have a question, Dr. Lee. So you mentioned
Christina.
One interesting fact near the end. You mentioned, well, there was a lot of them, but near the end, you mentioned how, you know, patients from different doctors' offices will have, from one doctor's office will have similar results, will look similarly. What do you think that's about? Do you think that's about the doctor's approach?
Yeah, I mean, you know, yeah, I think that's basically what it boils down to, Christina. It's all about their way of, of doing business, pardon the expression, but basically how they interact with the patient. And this is a fairly universal finding. So we always worry about that. It's also true within the institutions. I mean, the way they practice medicine as an institution, clustering. But the broader the cluster, in the sense of the people involved in delivering healthcare, the less the clustering effect manifests itself, because there's enough variability that kind of overwhelms that. Okay. Right. What clustering is kind of almost an elimination of variability. In the perfect cluster, everybody has the same response, right? And so depending on the individual or individuals that are involved in, let's say, again, delivery of healthcare within the cluster, whatever the cluster is, that dictates how reduce the variability or increase the correlation. That's really how you think about it.
That makes sense. Yeah, we want clusters to be alike, so it makes sense that we want to widen it to reduce that variability.
Right.
Thank you.
Okay, if there's